Hey guys, Mr. Hyatt here. This is part 4 and hopefully the last part of chapter 43, the immune system lecture from AP Bio. So we left off talking about antigenic variation. Here we're talking about latency. So some viruses may remain in a host in an inactive state called latency. HIV, for example, can be latent in someone for years. Herpes simplex can be latent in a human. You can carry it around without presenting symptoms. But once you're infected, you can never be rid of that virus, and outbreaks are always a potential problem, especially under stress. Anyone who has dealt with herpes simplex can tell you that around final exam time, they're going to have cold sores, and around big family events, they're going to have cold sores. So for a great case study here is going to be HIV. HIV infects helper T cells. Those helper T cells, losing them impairs both sides of the response. Remember, our helper T cells help everybody. So HIV attacks the molecule that helps both sides of our immune system work. Then it's going to evade the immune system because of antigenic variation. There's going to be a ton of mutations and the ability to remain latent while it's integrated into the host DNA. So we carry it around for a long time. Here's a graph that shows latency. So let's break these down. We'll look at the blue line first, relative HIV concentration. We start at 0. There's exposure. Quickly, we're going to have our primary response where we're going to build antibodies. Helper T's are going to diminish quickly, inversely related to the HIV concentration, because remember, HIV is breaking down helper T cells. Helper T cells get pretty low even when we're latent. So latency, remember, it's inactive, so it's just kind of riding around. Then AIDS actually develops once the helper T cells are pretty much destroyed. So that's kind of the difference between HIV and AIDS. There's a cutoff there. People with AIDS are highly susceptible to opportunistic infections. Most people die of some basic respiratory infection or other, like I said earlier, basic infections that most of us can fight. It's a worldwide problem. We've had some success with our treatments, but it is tough because the virus does mutate as frequently as it does. So we basically have to vary our drugs, stay in front of the mutations, and do some things along those lines to treat HIV. Another thing that should be popping in your head, I guess, at this point is cancer, because cancer is an issue where we have too many of our own cells. So can we manipulate our cells to recognize cancerous cells as non-self? There are two explanations for why frequency of cancers increase when immune response is impaired. The immune system can normally suppress cancer cells, or increased inflammation, the increased inflammatory response increases the risk of cancer. Cervical cancer that you guys have all heard commercials about for basically your whole lives, the HPV vaccine and all that kind of stuff. They can be prevented by vaccines because we ramp up our humoral response, and it sort of helps us with both of these. It kind of gears our immune system to fight cancer. So, in summary, there's a lot here, 4 videos worth. Try not to lose sight of the big picture because of the details. There certainly are a lot of details that you need to be able to regurgitate, for lack of a better term, but focus on this summary, and I think this will guide you to the depth that you need to know these things. Remember to look at your active words, your verbs. Distinguish is different than explain. Distinguish is different than describe. You need to be able to tell the difference between something describing an innate response and something describing acquired immunity. Know those 4 phagocytic cell types. Describe them. Talk about what they can do, all that kind of stuff. Distinguish between these terms. These are terms that lots of people mix up. So if I were you, I would be practicing on flashcards or T-tables or whatever you use for vocab. Practice, 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 practice. Explain, here are our bigger ideas. Explain how B lymphocytes and T lymphocytes recognize specific antigens. Explain why the antigen receptors are tested for self-reactivity. That's that apoptosis stuff that we talked about in the B cells and the T cells. Describe clonal selection. Make sure you know the difference between an effector cell and a memory cell. Remember, effector cells are causing an effect. Memory cells are remembering. Again, describe cellular basis for immunological memory, those T cells. Explain how a single antigen can provoke a robust humoral response. That's our clonal plasma cells, clonal reproduction, all that type of stuff. Compare neutralization and opsonization, similar but different. Describe MHC in rejection of tissue transplants, self-recognition, all that kind of stuff. I mentioned the different IG molecules, immunoglobulins. IgE is the big one. So if you only learn one, learn IgE. Describe what happens during an allergic reaction. You may have an essay question on this on your test. Describe some of the mechanisms that pathogens have evolved to thwart. How do pathogens stay ahead of us? List strategies that can reduce the risk of HIV transmission. I didn't talk a lot about that in the video. Pick that up from your book. You know most of those. And then make sure you're using your book. Look at the concept check questions in the textbook. Look at the before and after questions. It's probably a good idea to sketch out some of these ideas in terms of like the positive negative feedback, the antagonistic relationships. Those are probably all really good ideas to work out in your head, so I don't surprise you with something like that on the test. So that is finally the end of chapter 43.